Good morning, everyone. Good afternoon. My apologies. I'm having some technical problems here in the
Hey, Eric.
Silicon Valley. Good to see you all.
Nice to see
You have a
you.
great camera.
What's that?
You're looking like you have a great camera. You look like Netflix movie,
Exactly. Whatever will help to make me look young.
You look younger and younger.
Just got to try to convince you that whatever we're doing is actually working.
Yeah, right.
It's good to see all your faces. And so
Eric, we've got a couple folks that are just logging on now, and so we'll just get 1 second before we start.
What we thought we would do today is two things. One, I want to sort of bring you up to date on some of the new direction that the Institute is taking in terms of human biology, and really give you an idea. We've had a number, two big recruitments in the Institute in the past 2 years. One was Lee Hood, and the other one was Nathan Price, who are both true pioneers in terms of what they're doing. When we recruited them, this was, just to give you a little bit of sort of historical perspective, this was a bit of a leap of faith in a way that no one in the aging space was talking about doing phenomics of aging. And it was a bit of a hard sell here at the Institute with some of our faculty in terms of, is this really the direction that we should be going on? But we persevered, we got them on board, and what's really exciting to see now is that the whole field of aging is actually moving in this direction. And I will take some of the credit in terms of having really pointed the way. And I'm now part of a big biomarker of aging consortium, which is what this is all about, essentially, is, how do we take all of the discoveries that we've made in the aging field in animal models, how do we translate this into humans? And humans have, as a model system, has two big problems. One is safety. If you're thinking about interventions that are you expecting are increasing lifespan or health span, you want to make sure that they're actually not doing the opposite. And we know any interventions, any drug has the potential to actually make the situation worse, especially when you're thinking about an intervention that's going to be administered for many years. So that really is the biggest problem of anything related to aging in humans, is you want to make sure that whatever you're doing is 100% safe. Don't want to find out 10 years from now that the buck has recommended this intervention or this drug, and that people are actually living shorter. That would not be good messaging. So the bar is much higher than if you think about a traditional drug development against cancer, where you have a patient who has a limited lifespan of 3 to 5 years. If that patient is not treated, and you gain an extra year, wonderful. So that's the easy part. The aging space is really confronted with something much more difficult. So number one, safety. Number two is we are long-lived animals. And how do you, whenever you start an intervention, how do you actually get to the point that you can predict within a reasonable period of time? We don't want to do 30-year clinical trials. How do you reduce this long life and be able to measure something in a shorter period of time? So this, out of these two needs came the whole field of biomarkers of aging, which right now is one of the most rapidly growing fields of aging research, and we're among the leaders in this area. There are I would say three organizations in the world that are leading the way. One is Harvard colleague Vadim Gladyshev. Another one is Mike Snyder at Stanford, and the third one is the Buck. And I'll tell you a little bit towards the end of the presentation about some of the efforts that we are undertaking really in this direction. There's some new grant application with a program called ARPA-Age, will allow us to hopefully get additional funds. So do we have everybody on board? Brian, do you want me to get started?
Yes, I think now is a great time to start. That'd be great.
Okay. So I have a few slides that I want to share. So one thing that I've heard from the group and is that many of you are interested in sort of a going back and forth discussion. So I will try to keep this presentation kind of short, and then hopefully we can chat. The agenda is pretty loose in a way that I want this to be answering some of your expectations and interests. So it can be a mixture. I don't want to be speaking for an hour and then see you in a month or two. So I want to make sure that most of you feel that you're getting something interesting out of it. So let me share what we are, this brief presentation about.
And while Eric's pulling that up, if you have questions you want to ask, you can raise your hand, or you can also put something in the chat, and we'll make sure we get that up to Eric.
Okay. Thank you.
One thing we could do also is a hybrid, where from time to time we simply have a meeting with your questions, and we can, if you want to submit the questions that you have to Brian, we can curate these questions, try to see, which questions are generating the largest amount of interest, and then try to spend some time addressing them.
Eric, the screen we're seeing is your email. I want to switch. There we go. There, good.
You seeing me now?
Yep. We're seeing the slides.
Okay. All right. Is it full screen?
Not yet.
Oh. Use slideshow like this. Is it full screen now?
Looks like it's loading.
Loading?
Yeah, it's just black right now.
How about this? Something is not working. So what do you see now? My email?
Yes.
But I'm really completely confused. And how about now?
We see the slide deck. You can just walk through it. I don't think you need to do full. For some reason, we're only seeing the, oh, there you go. That might do it. There you go.
There it is.
Okay, now you have the full slide? Okay.
Yes.
All right. Okay. So there's something really screwy the way it's working now. I don't know why it's doing this. Okay. So a groundbreaking new look at humanity. This is nothing less than this, what we're proposing. So you've all heard about the human genome, which was sequenced for the first time, using technology that actually our own very own Leeuwenhoek designed and invented. That was completed in 2000, and it led to a whole series of companies proposing to use this type of genomics data to predict health. And you've all heard about 23andMe and others. The data has been disappointing in a way that genomics is important. It will identify a number of diseases, but in terms of prediction of value and your health, it's been somewhat disappointing. And the reason is the following. It turns out that about only 97% of your lifespan and health span is determined by genetics. So by what you've inherited from your parents. And about 93% is non-genetics, which would mean lifestyle factors. I mean, this is disappointing for the geneticist, but it's good news for all of us, because it means essentially the way you are going to be aging is determined by how you live. And that includes, your environment, but also includes what you eat, sports, all of the things. That means essentially for most of us, your lifespan and your health span is under your own control, as much as you can actually control it yourself. It turns out that there's an exception to this, which is the families of, there are a number of long-lived families where there are abnormal numbers of centenarians. So if you have a centenarian in your family, your first-degree relative, that's a centenarian, you are, I think, 90% more likely to live above 90. So there are definitely some families where there's a stronger genetic component. But for most people, really, your life expectancy is non-genetic. So the question is, knowing all of this, now how do we actually measure health? We've talked about this idea of generating these biomarkers of aging. What are they going to be looking at? You all know about DNA, which generates genomics. Much of the analysis that we have been able to do is based on the idea of measuring simply your genome. So it turns out there are many other things that we can measure. As I've just told you, just your DNA, which in some ways is immutable, does not provide that much information. We're at a stage now where we can measure RNA. This is called transcriptomics. DNA is transcribed into RNA. RNA is used to make proteins. So measuring all of the proteins is called proteomics. And these proteins are involved in managing a whole series, hundreds of thousands of different metabolites. And so measuring these metabolites is called metabolomics. Today, in terms of research tools, we can measure all of this. In the clinic, we don't measure any of these, which is remarkable. So the technology is such that I can measure out of your blood, I can measure your transcripts, I can measure your proteome, I can measure your metabolome. This allows us to generate hundreds of thousands of data points. And so right now, there's a big discrepancy between what we can do in the laboratory setting and what we can do in the clinic. Now, what are these data from? The thinking is that the more information we get, the more likely we are able to make a correct decision. If you are, right now, if you go to your physician, he or she relies on maybe 100, 150 data points, your LabQuest or your LabCorp or Quest lab results that you get, you can look, there's maybe 100 different values. There's a huge discrepancy between the simplicity of this and what we can actually generate in terms of data. So there's the assumption that we are working on is that by getting more data, we're going to get more reliable and more precise information. And there's a good example of this that I'm showing you on this slide. Everyone knows about the ApoE variant. Each of us has two variants of the ApoE genes. It can be E2, E3, or E4. The E4 variant is known as a genetic risk factor for Alzheimer's. So if you are E4, you are significantly more likely to get Alzheimer's disease. But this is very strongly determined by other factors as well. So just your E4 status does not tell you the whole story. And then what I'm showing you on the right is what happens if you're Hispanic versus Black versus white versus Japanese. It turns out if you are a Japanese and you're E4, you're 35% likely to get Alzheimer's. But if you're Hispanic, you're only 2%. So this is really a good example of sort of multigenic type effect that we should be able to measure. And so this is an extreme example. There are many others. So each variable by themselves carries little weight and has to be put in the context of all of the other things that we can measure. So this leads me to the big question that we're trying to address is what data are necessary to understand health span and lifespan? How do we measure them efficiently? And so this is one of my favorite pictures. Really, if you want to live old, obviously, it turns out that many of the existing centenarians actually have a very unhealthy lifestyle because they have such a strong genetic background that protects them against anything. So talking about the phenomics of aging, phenomics refers to all of these omics, all of these markers that we can, transcriptomics, metabolomics, proteomics that we can generate. So this is a whole new program that is started at the Buck about a year and a half ago, was inspired by the recruitment of Lee Hood, and I've been working really closely with him to really implement this into the clinical realm. So we went already very quickly in terms of what is the phenome. So the phenome is your genome, that's at the top left, but it is also your many other measurements that can you see my mouse, my arrow? Does it move around?
Yes.
Yeah.
Okay.
Oh,
So
yeah.
if you look on the blood, clinical chemistries, metabolomics, lipidomics, proteomics, immunomics, but this is what we've talked about. But it is a lot more than this. It is your social determinants of health, what kind of an environment, how pure is the air quality? Do you have volatile organic compounds in your house? Are you, because of your work, exposed to a number of potential toxins? It includes, self-tracking. The wearables is one area that I'm really excited about because it turns out, I don't know how many of you on this call actually are wearing a wearable. That could be the Oura Ring or the Whoop or an Apple Watch or a Fitbit. These technologies are exploding right now in terms of their capabilities, but also in terms of their predictive value. I sit on the board of Whoop, the scientific advisory board of Whoop, and without divulging any kind of trade secrets, I can tell you they are doing things that I would say will blow everybody's mind in a couple of years in terms of what they are able to measure and predict using a relatively small number of variables. And I predict that in the long run, we will all be continuously monitored by these agents, and your physicians will have access to the data and will be able to integrate this into the full picture of your health. Because it turns out, if you think about our model of medicine right now, essentially it's sort of point of care. You go to see your doctor once a year, you get a measurement done on one day. And from this, you're trying to infer what is the life of the patient for the rest of the year. Well, it turns out from the wearables, you can pretty much measure a lot and predict actually a lot of issues related to healthcare and sick care and so on. Okay, so the phenome is essentially the idea of integrating everything that you can about a patient's health. And I mentioned also here the microbiome in the lower right. This is really analyzing the bacteria that are living in your gut, in your skin, in your mouth. Each of these distinct bacterial colonies play a critical role in our health. So again, this is the point that I made earlier. When you look at typically the way we practice medicine today, you have your clinical blood test, you get dozens of data points, your complete blood counts, your cardiometabolic panels, and so on, versus what we can measure today. Your blood proteome, we can detect at least 3,000 proteins, lipidome, 1,000 different lipids. These are fat proteins. Digital health technology includes hundreds of measurements, your microbiome sequence, 10,000, your whole genome sequence, and so on. Just imagine the amount of data that we have at our disposal. So how do we go from left to right? And this is what we are trying to build. So here's where the promise of all of this is. If you are a patient and you are well, you might have a disease that is taking place, and this is illustrated here by the little green dots at the bottom. So this, what I'm showing you here, is a network. Imagine this network could be a whole series of proteins or cells in your body that are functioning. So you have a disruption of this network, something is not working, a protein is lacking, or so on. And this is happening way below the screen, so you are not aware. This disease is going to slowly progress. And by the way, you are still completely asymptomatic. You have not noticed anything. All of a sudden, there's going to be a first detectable clinical marker. So out of this whole disrupted network, because we're measuring such a small fraction of it, we're going to see, the clinical diagnosis is going to happen at a time which is very late. And I'll give you a good example. Many of you go see your doctor, you get your blood sugar measured, and your blood sugar, if it's below 100, is normal, 100 milligrams per deciliter. If it's above 100, you have prediabetes. It's above 120, you have frank over diabetes. Now, we know today, when we look at the network below the blood glucose, that 15 years earlier, we can already detect these green dots here on the left, okay? So it stands to reason that once you have a clinically manifest disease, as you see here with the first detectable clinical marker, this is a disease that's been going on for a long time, and you're looking at the end result of a process that's been ongoing for 15 years. Now, this is what medicine does today, okay? It will start treating you when the whole system essentially has gone awry. You're detecting the clinical marker, but the whole network below is already disrupted. So phenomics medicine is very different, as I'm showing you here. What it will do is 15 years earlier, we'll be able to first detect some phenomic changes, the little green dots, by measuring all of these variables, will allow you then to target a therapy. One of the things that's important to realize is if you diagnose disease at this point and you start treating, you don't know where the primary lesion is. So you're treating essentially the symptoms, and you do this with very little efficacy. You know, if you are diagnosed with type 2 diabetes, you're going to go on metformin, and only about 30 to 40% of people will actually respond. Most will not respond because it's in some way it's too late. So the idea of targeted therapy that really target the primary lesion and goes in early is going to be critical. Now, hopefully, if you do this type of therapy, you're going to be able to return to wellness. And so this is really the whole. This slide, in some way, for me, is really encapsulates the whole wealth of what we're trying to do. Essentially, preventing disease before it gets to the stage where it is too late to really revert in a truly effective manner. You know, this model, by the way, has been validated in animal models, so we know we can do this. What's really interesting is some of you might have, I don't know, I suspect some of you are on the statins, which, you know, lower your cholesterol. You can imagine your cholesterol level, again, being one of these clinical markers that is kind of late. It turns out that when you look back at some of the early lesions, you find some things that medicine has already discovered in some cases, like the PCSK9 inhibitors. Some of you might have heard about a drug called Repatha. PCSK9 seems to be one of these really early modification in heart disease that can be targeted for a complete reversion of disease. Okay, so how do we do a phenomic study? Essentially, you know, we start with a large number of participants. You sample biobank. You conduct all of these multi-omics analysis, digital analysis, clinical markers, self-reported analysis. And then there's the core of all of this is, because now we're dealing with hundreds of thousands of data points, is to integrate all of this using AI. You know, medicine is very good at what it's doing because we have very small number of variables which allow you to make inferences. When you're dealing with hundreds of thousands of data points, no one can actually really analyze it. So using AI, we can now identify early disruption patterns and so on. And then this will lead, you know, in the future to obviously new knowledge, new therapies, and so on. So now the good news is that we already know, at least Lee Hood and his colleagues have already established a proof of principle. There was a company called Arivale where they conducted this type of analysis on 5,000 individuals. This was a commercial company which did not survive for a number of reasons. Sometimes you can be a little too early in what you're trying to do, but we all recognize the value of this. So in this analysis, they did exactly what I told you, this deep phenotyping, including many digital measurements and brain assessments and so on. And the good news is that they were able, in these people, this is data that shows that this personalized intervention improved biological age. Even within, you know, a couple of years, they were able to reduce the biological age of these people. They made them age slower. So enormous potential in the future. So when Lee joined the Buck and Nathan Price as well, who actually has worked for many years with Lee, The first thing we are aiming for, we just obtained a very large grant called PATH, which is up to $52 million, which will allow us to essentially really take the Arivale approach and really put it on a much larger scale. The first thing that we want to do is to understand circadian dynamics. So what do I mean by circadian dynamics? As we are changing constantly throughout the day. And I just need to give you an example. And so biologically, we are not the same person in the morning as we are at night or in the morning. The best example of this is that we all know we're sleeping during the night, we're fasting during the night, but there's a lot more that can be measured. And it turns out that the circadian rhythm and how well you are aligned with the circadian rhythm has a very profound effect on health. For example, we know that the time of the day of surgery has a measurable impact on mortality for surgery. There's, peak areas where we know there's more mortality on a global scale. We know, for example, also your immune function, your gene expression, all of this is circadian dependent. You might know, for example, that your cortisol levels increases in the morning. That is the hormone that gets your body ready. And that makes you somewhat insulin resistant. This is why I tell people, should not be eating sweets for breakfast. That's really one of the number one rules. Morning is a time where carbohydrates are not metabolized well by your body. We also know you've all experienced jet lag and what it means in terms of sleep, in terms of appetite, in terms of mood, all of this. We know, for example, that shift workers, people who are constantly subjected to changes in terms of the hours of the day when they work, is a high risk factor for early mortality in the long run. So the question then, what happens to the phenomics of your circadian phenomics? Yeah, shift work is a carcinogen. This is another way of saying the same thing. So the first study we're going to be doing is, as we are going to be measuring all of these variables in people in clinical trials, obviously it's not always going to be at exactly the same time of the day, although one could argue that it should. But the first question we're going to be asking is taking 125 individuals over the age of 55 and to really deeply characterize them. So this is called the TIME study. We're about to start. So we're going to be we have a number of volunteers who have joined us. We're going to be studied for 10 weeks, and they're going to be getting a lot of measurements. For example, you look on Thursday, 5 time points for omics, 8 a.m., 11 a.m., 2 p.m., 5 p.m., 8 p.m. Again, repeat it Thursday, Friday, Monday, and Tuesday. What these people, so this is going to allow us to determine really how much does it vary from first for a given individual during the day, but also between different days. Are these data reproducible, or are there some markers that are more fluctuating that, for example, that might be more related to what you're eating and so on. So really, this is it seems like a very basal type of study, but it's the foundation for much of what we're going to be doing in the future. We're also going to be challenging some of these participants using a number of control perturbation. Some of them are going to get oral glucose tolerance tests. This is a bolus of glucose, trying to see what happens in terms of their numbers. Some of them are going to be stimulated with a VO2 max test, essentially meaning getting on a bicycle and bicycling at maximum strength for close to 30 minutes. And so we're going to be able to see what is the effect of exercise in a very basic way. So this would be one of the early study that's going to be foundational to many of the other things that we're going to be doing. We're also very interested in a female-specific biorhythm. Men are a relatively simple organism in a way that we go through the year pretty much in the same way. We all know, menstruating females have a 30-day cycle during which the woman's biology really changes dramatically from being before ovulation, during ovulation, after ovulation, and during the menstrual period. So this is a data desert, believe it or not. This has not been studied. We do not know what happens in terms of the biology of women. Now, this has profound implications. We know, for example, that women respond to fasting very differently than men. Can we measure this? Can we identify what is really different at different moments in the menstrual cycle? We also are very interested in, what happens during menopause, premenopause, and after menopause, the whole sort of perimenopausal period. We know that there are dramatic changes in the way women age before and after menopause. So we want to measure all of this, really trying, the term data desert that is listed here is not an exaggeration. Despite everything we know, there are some areas that have been not receiving the attention that we should be focusing on. So, and this is part of a whole new center at the Buck called CHARS, Center for Healthy Aging in Women, which is going to be collaborating with the Center for Healthy Aging at the Buck and really trying to address specific issues related to women. I think I will stop here. This is a lot of information. I just wanted to give you a broad idea of some of the big directions in which we are. And so this leaves us another 30 minutes for me to answer questions or that can be related to this or to anything else that you are interested in.
We have a couple of questions that were submitted ahead of time, but before we get there, is there anybody that has any questions about the work around phenomics or what we're doing with the Center for Human Health Span and the PATH project? Anything, Andy?
Yes, Brian, thank you. Can you hear me okay?
Yeah.
Yes.
Great. Eric, I'd be very interested in learning from you how much in the future our new insights into how to address aging are going to come from the clinical studies you talked about, which are clearly very important, and how much, on the other hand, it's going to come from computer modeling and from computer data simulation of the human body. And I know this is an area that some of the Buck staff are working on. And it's unclear to me where the trend is as we look out over the next 5 years. Could you address that a little bit?
Absolutely. This is something that it's sort of a pipe dream that's been there for biologists for ever since I started. I started at the University of Brussels, and one of the colleagues next door, this was in 1984, had spent his whole life, which is a 40-year career, trying to model genetic pathways and genetic regulation. So people have been working on this idea of modeling using computer systems what happens in biology. And even then, people were able to model, for example, a simple genetic pathway in bacteria and to predict exactly what would happen. Now, going into humans is a whole other, going from a bacteria and mapping one metabolic pathway. For example, there was a famous Nobel Prize winner by Jacob and Monod, two French scientists who got the Nobel Prize for discovering something called the lac operon, which was the first fully understood genetic pathway where they could show that the abundance of a metabolite was triggering a response in the cell that activated an enzyme that metabolized this metabolite. So this was, a really simple response where you have something increases, it triggers the activation genetically of a protein that goes digest. It goes down, the level of the protein goes down, and the gene is turned off. So that's a lac operon. Nobel Prize winner, by the way. Very easy to model. When you move, and so there's been a dream always in terms of biology to really put a big model system that will allow you to predict what would be the outcome. And I think, there are multiple examples. This whole field now has really been accelerated dramatically for two reasons. One is our understanding of biology is much deeper. Okay, so if you only have two variables, you can model it, but obviously you don't model the whole organism. So increasing the knowledge and the understanding of all of the genetic pathways is there. Not complete, but it's there. Our computing power is also increased dramatically. And so those two have gotten to the point where we can build a much more complex model and much more predictive model. And so this whole field is called the digital twins. And it's now moving into humans. So this by the way, Nathan Price is one of the world's whom we've just recruited is one of the world specialists in terms of these digital prints. For example, he has built, so let me just clarify, we don't have a model of even a single cell because that the complexity is just bewildering. I'll just give you an example. When we talk about building a digital twins of humans, we have each of us 14 trillion cells, okay? 14 trillion.
Wow.
Now, today, our most, just even to think about our brain, because there's been a lot of talk about trying to model the human brain using artificial intelligence and so on. Our most complex computer system today, in terms of the number of connections, if you were to make the equivalent number from our brain organization to a computer, our most complex computer system is about 100-fold more simple than the human brain. So even though we're getting to build very intelligent AI-based system, we still, in terms of the potential of connections, 100-fold lower than the human brain. But that's growing very rapidly. It's predicted. When people talk about AGI coming up in the next 3 to 5 years, this is essentially our computer system having the capability of connection that the human genome. So anyway, I digress. So these digital twins are rapidly evolving. One of the applications that I know that Nathan has built, and I'm happy to have him come and tell you about digital twins the next time if you want.
Yes.
I think that'd be interesting. I know he has a model system, for example, that predicts response for Alzheimer's disease. And that's based on a number of variables that essentially reduce a very complex system into maybe 100 different variables. So I think this is a field that is going to grow in relevance and importance in the future. And it's one in which we are working on.
Thank you.
Yeah, Alan, do you want to unmute?
Hi, Alan.
Hi, Brian. Thank you for the invitation. Hi, Eric. I had a similar question. I just wanted to volunteer our expertise. I run a research lab, and for the last 15 years, we've been building very, very complex digital twins of full supply chains, all the way from mining to the retail outlet and everything in between. And I see so much similarity in building these models for biological systems because you have to select the level of abstraction. So I'd be absolutely happy to volunteer our expertise and see if there's some commonality. We've also built very advanced digital twins of the supply of pharmaceuticals and medicines to get it into areas to prevent shortages and surpluses at the back end. So just also wanted to find out if you are active in that. It sounds like you are. And I think that there's a lot of shared learnings that we can apply to what you are busy working on.
I love this, Alan. Thank you and we will be following up with you. In some way, when we talk about these digital twins, the idea is computer modeling, and which is obviously not only applied to medicine, it's applied to supply chain, as you said, to climate change. All of these things are in some way digital twins of phenomena that occur all around us. The problem we have in terms of human biology is the sheer complexity. It's bewildering when you're thinking about 14 trillion cells that are all interacting with each other. But we will get there. I think I'm convinced. And obviously, the key is at this point is to identify which are the variables that are the most predictive of the model. Sometimes I had a little bit of a cynical approach to the whole field because it turns out people are using variables so that their model fits with reality. But then, we have reality, which is already better. So, but right now with AI and all of this, I think I can definitely see the field moving very fast and learning from people, from industry. I imagine, a company like Amazon, I mean, I know is all digitized in terms of consumer behavior. They can predict, so much in terms of what's going to happen, when a surge in the demand of a given product is going to happen. So I think so we can definitely use the health.
Yeah, I'm an advisor for Amazon. They process 450 packages per second through their supply chain in the United States. And I think two things that's interesting was we initially started thinking that you'd have to know everything about every part in order to predict emergent properties of the system. And it turns out that you don't. You need to find out to what level of abstraction to measure the part to actually predict quite accurately these emergent properties of the system. And as you mentioned, yes, there's exponential growth and complexity as you add more parts and interdependencies. But again, with the technology and the AI side, it's very powerful. We can use the digital twins to generate a lot of synthetic data that the AI can train on. And once the AI has been trained, you can then validate those hypotheses against traditional views inside of the digital twin to see which one will give you the best long-term results. So yes, just again, to volunteer my services, I'd be happy to work with your team on this.
Love it. Love it. Thank you.
Before John had submitted a question, but before we get there, we had a number of folks in advance, Eric, who were just wanting to get an overview and just sort of a big picture look at your thoughts about GLP-1 and GLP-1 medications and how they might be influencing or impacting the field of healthy aging and longevity.
That's a tough one. But I'll do my best. Where do we start? So first, I mean, really what is known is that if you give these people who are this drug to individuals who are overweight, they are incredibly efficient at reducing weight by 10 to 20% over a few months period. So that the efficacy is just remarkable. What has been discovered is that people are not only losing weight, but there seems to be a whole series of biological effects that were not expected. We see increase in kidney function. We see increase in brain function. We see in cognitive tests, we see increase in heart function. So there seems to be a whole, and I don't know, let me see if I can pull up a slide to show you one of the paper that was published recently that discusses this, or I can maybe share it with you for you to take a look. Where would this be? I did not have this ready with me. I'll have it for the next time, and I can.
Yeah, and I can send it out afterwards.
I can share it with you. So remarkable effects in terms of health. Now, the key question. So one of the obvious interpretations. So the drugs does two things. It lowers your weight, but it also increases your metabolic patterns in terms of glucose sensitivity. We see people who are diabetic or pre-diabetic. We see their numbers are being normalized. Now, all of the other health effects that I've described, and they're really impressive, could occur for two reasons. One, the drug simply lowers your weight and improves your metabolism, and this is enough to make you, every one of your organs actually function better. So what we have not been able to do is to decide, okay, how much of these effects are truly anti-aging effect, because the drug behaves like an anti-aging drug today. How much are these caused by this versus how much are these effects the consequence of the fact that you've lost weight, basically. Now, and this is in some way, this is the whole discussion that is taking place. the secondary aspect of that question is who should be on these drugs? Is it only for people who are grossly overweight, or if we see these enormous effects on them, will we see effects on people who are slightly overweight? Okay, and which obviously has enormous public health implications. And I would say that I lean on the side that these drugs are going to have profound effect on most everybody unless your weight is perfect. And so this really calls a whole series of ethical and questions for people to ponder as a society. And for all of you, if you find yourself to be a little bit on the side of being overweight, should you be on the and I get a lot of people asking me this, should I be on the drug if I'm a little bit overweight? And I can share, one thing I can share with you is personal experience, because I've been on it now for 6 months, and simply because I was curious, and also because I do self-experimentation, and I think a lot of us in the field are testing these drugs and really trying to see what does it do before I can share this experience with you. Now, my personal journey with this has been that I was 187 pounds when I went on this, so BMI 25, which would be considered normal for someone my age, actually even more on the lower side. And I've taken the drug it's called tirzepatide since June, so that would be about 7 or 8 months. I have lost about 12 pounds without trying, which is actually, it's been pretty remarkable. No loss of appetite, sort of nothing, so 12 pounds lost. My numbers have improved in a way that's been pretty shocking. Fasting blood sugar went from 95 down to 85, which is really sort of like the blood sugar of a 20-year-old. My insulin levels have gone down. We know insulin levels are highly predictive of your metabolic health. Right now, my fasting insulin is extremely low. No loss of muscle mass. This is another thing, whether the big bugaboo that's been advanced by people in terms of saying, well, these drugs induce more muscle mass and therefore they're bad. It turns out that, by the way, what I'm taking is what would be considered a microdose. It's a really small amount. Starting, your typical patient on some of these drugs will take up to 15 milligrams. They start at 2.5 milligram, like they call this the adaptation dose for a few weeks. I'm still at 2.5 milligrams, so I'm at the starting dose. I've never taken higher. And despite this, I've seen dramatic effects in terms of my biology. So I would argue that the way medicine right now is using these drugs is they are inducing weight loss, which is too rapid. So people, have these dramatic weight loss over short periods of time. And we know that by itself is sufficient to lose muscle mass. I recently saw data from a colleague that illustrated the fact that the loss of muscle mass during weight loss is universal. It's age dependent. So the older you are, the faster you lose weight, the more you're going to lose muscle mass. In my case, I do a lot of sports. So I think I have, I did not do anything special, but I've lost 0 muscle mass, but I've lost significant amount of fat mass. And right now I'm at about 9% fat mass, fat body mass, which by all accounts is somewhat in the optimal zone. So for me, the experience has been remarkable in many respects. I don't know if I'm going to stay on it. To be honest, I would be tempted because it just has solved so many issues. For example, I'll give you one example. I'm one of these people that never felt satiety. And I don't know if some of you, this can resonate with you. I always had to stop eating because I realized, okay, if you eat too much, you're going to gain weight. and I know some people who would be at dinner looking at their plates and saying, I'm full. This never happened to me. I was never full unless Thanksgiving, I would be full. And you know, after having been on this drug for about 6 weeks, I had this first experience. I just looked at my plate and said, I can't eat anymore. I'm full. I was just not hungry and not wanting to eat. So this is now there's a lot of debate, you know, and we're going to hear from the new administration that, you know, putting people on everybody, the whole country on drug is just not the way we should go. And I, frankly, I agree. Until we reform the whole offering of what is available in our supermarket and so on, if we are evolutionary misaligned with our environment. So we have evolved as an animal species to do two things. One is to be always hungry because when food was available, there had to be a very strong impulse to grab that food, eat it, and to transform it into fat. You know, this idea that food for most of our evolution was a commodity, something that when you found it, you had to have the immediate desire to eat it, and once you ate it, to immediately transfer it into fat. So this is our biology. Now we live in an environment where the food is abundant and is constant and is actually, frankly, has been engineered to be even to really call on to all of these addictions. So, you know, we love sweet. We love sweet because the body recognizes this is a very quick way to store energy in terms of fat. So the idea that, you know, and this environment, which is highly toxic, by the way, this is not an American specific process. The whole world is becoming obese with somewhat of a delay, you know, in some countries, but except for Japan, which is a remarkable example of how you can actually engineer this at the society level. And I've been reading a lot about what Japan is doing. It's absolutely amazing what they're doing. They have 4% obesity. We have 60% obesity. So there's a lot to be learned from the Japanese, but they're the only ones right now. Everywhere else in the world, obesity rates are climbing. And I was just in the Middle East. They actually are even worse than in the US. So how do we deal with the situation like this? I think we need to reform the whole, you know, food industry, but we also maybe we need to help people to get back to a regular weight. And my prediction is that once you get to a more healthy weight, it's easier to exercise. It's easier to make the good choices in terms of eating and all this. So anyway, so this was a very long question to a very short, a long answer to a very short question, but I think I've really, it's been an amazing experience for me to go on this. The question is, I'm tempted to stay on it. I've also measured, as you know, I measure all of my clocks. Everything is trending in the right direction. I haven't seen any evidence of any sort of negative side effects. It's low dose. It's slow weight loss. I mean, I've lost 1 pound per month, if you think about it. It's really not a rapid and drastic weight loss. I would argue that unless your weight is perfect, many people would benefit from this in terms of their health in the long term.
Eric, we're about ready to close, but Claudine had one quick question, and maybe we can just deal with the first one, which is, has there been any evidence of women having a similar effect, or the drug having a similar effect on women as it does on men? Is there any gender differences?
Not that I've seen. Not that I've seen.
And is there a minimum or ideal age that people might want to consider? I'm not sure that approaching maybe a microdosing protocol.
It would obviously depend on, you know, level of physical activity, you know, level of obesity. I mean, frankly, knowing how these drugs work and the effect that they, you know, one thing that should be interesting to note is you still have to watch what you eat. I still could overeat, and I mean, it does not completely replace the idea of, you know, eating sensibly and all of this. But it certainly is an amazing help. One of the most amazing things that has happened to me is I have a very sweet tooth. And this has turned it off completely. I can really look at a pastry and say, no, I don't want to eat this. It just, and there is a worry right now. The same thing is people have seen the same decrease in interest in alcohol and in some of the other, the fast food type of food. And there's a lot of worry right now in the fast food industry because people are just losing their appetite for these types of food. So the drug, I think, seems to be having an effect on the brain in terms of these reward circuits, which, by the way, are the same for drug addiction, for alcohol dependency, for food dependency. So these are all, we know, glucose is addictive, just like cocaine. The more you eat sugar, the more you get addicted to the high that it generates. So the drugs, I think, might be targeting some of these reward pathways that are central to many of our behaviors, basically. So it's really, as an endocrinologist, as a scientist, as a physician, as a self-experimenter, I have never seen anything that excites me more in terms of potential for human health.
Quickly, Eric, what happens when you reach your ideal weight? Would you continue to take it?
That's actually, I'm getting to the point. I had one person yesterday who looked at me. She said, "You lost too much weight. You don't look healthy." I said, "Well, it's because we assume, to be plump and to be a little bit over padded as being healthy." But I would argue, I argued with her. I said, I have 9% body fat. I have a good amount of muscle. I have more energy than I've ever had in my whole entire life. So maybe I don't look healthy because, there's something called the Ozempic phase where people lose weight in their face. It makes you look a little gaunt and a little drawn. But I'm sure with time, this will rebalance itself. Now, the question from you, Lee, is a good one because that's one that I'm pondering. What do I do next? People have shown that if you stop it, people tend to gain weight. The question for me would be, could I find sort of a maintenance because I don't want to lose more weight. I think I'm at a weight where I think this is totally fine. I can even regain a little bit more muscle mass. But could I find a dose that would actually allow me to maintain this without any weight loss? That's the next stage. I'll tell you maybe 6 months from now.
I'll join you there. I'm underweight.
Well, we're going to stop here. I can't thank you enough for joining and for sending in your questions. A couple of quick things. I want to thank Robert. I think it was Robert Wall who suggested that we create a WhatsApp group. So we're in the process of doing that. So it'll be a private WhatsApp group where we can continue the conversation and dialogue, and we can update you easily in between calls. We're also putting together a reading list, curated reading list that we'd recommend, and that'll be part of that. And our next call will be in May on the 22nd, so be looking out for that. In the meantime, send your questions or any follow-up to me, and we will continue to keep the flow of information coming back to you. And we'll send something out in March just as kind of a follow-up. And also as new articles, new publications become available to Eric that he's interested in and thinks you guys should know about, we'll provide links and updates for those as well.
Cool.
I want to finish by thanking you all for being there. I think, at least for me, these are fun, and I look forward to doing more of them. Would also love to really, have you help us to build those the way you find them the most helpful. So we're really looking for your feedback, and please bring your friends. I've told Brian, I said, this will be successful when we have, a couple of hundred people that we can interact with and create really a whole community of people who are fascinated. I could imagine we could have an annual meeting where everybody gets together and talk about science and so on. So for me, it's a pilot. I told Brian, we're going to make it happen, but we need your help in some way, being the precursors, being here early with us. We can make it really what you think would be the most attractive to you and to potential friends and acquaintances.
Good to know.
Great.
Thank you all for attending, and recording will be sent shortly. And again, keep your questions coming, and we look forward to seeing you again soon.